Okay, for the third part, we're going to look at protections of the brain. So when we think about protecting the brain, and this also can go for the spinal cord, there are three features within the protective shell of the skull that provide additional shelter for the delicate brain tissue. The cranial meninges, these are three layers of membranes that surround the brain. Cerebral spinal fluid, which is fluid that bathes the brain and then fills the cavities, and then a structure or a functional structural area called the blood-brain barrier. This prevents many substances from entering the brain and its cells from the blood. The cranial meninges are composed of three protective membrane layers composed mostly of dense, irregular, collagenous tissue. The structural arrangement from the superficial to deep is the epidural space, the dura mater, and the subdural space, the arachnoid mater, the subarachnoid space, and the pia mater. So your actual structures are the dura mater, the arachnoid mater, and the pia mater. The dura mater being the outermost layer, the arachnoid in the middle, and the pia directly attached to the brain. The epidural space is between the inner surface of the cranial bones and the outer surface of the dura mater. It's only a potential space as the dura is normally tightly bound to the bone, only allowing for passage of blood vessels. The dura mater is the outermost meninx. It's the thickest and most durable of the three meningeal layers. It is a double-layered membrane composed mostly of collagen fibers with a few elastic fibers. There are two layers of the dura mater. The periosteal dura, this is the outer layer. This is attached to the inner surface of the bones of the cranial cavity, and it functions as periosteum with extensive blood supply to the epidural space. The meningeal dura, this is the inner layer. This is avascular, and it lies superficial to the arachnoid mater. The dura is mostly fused, creating a single inelastic membrane, except with regions where the cavities, called dural sinuses, are located. We'll define those. The dura sinuses, these are venous channels that drain cerebrospinal fluid and deoxygenated blood from the brain's extensive network of veins. So you have cerebrospinal fluid, which is providing the oxygen and nutrients, but then as that exchange occurs and you start to have CO2 and waste products, that will then be drained through the venous system of the brain, and that will go through the dural sinuses here. The dura mater also contains what we call dural folds. This includes the falx cerebri, tentorium cerebelli, and the falx cerebelli. The falx cerebri is within the longitudinal fissure. It forms a partition between the right and left cerebral hemispheres. The tentorium cerebelli is partitions between the cerebellum and the occipital lobe of the cerebrum. And the falx cerebelli is partitions between the left and right hemispheres of the cerebellum. The subdural space, this is a serous-filled fluid space. It's found deep to the dura and superficial to the arachnoid mater. It houses veins that drain blood from the brain. The arachnoid mater, this is the second meningeal layer that is deep to the subdural space. This is called the arachnoid mater because it's a very thin layer and it actually looks like a spider web. This particular web-like membrane is composed of dense, irregular, collagenous tissue with some degree of elasticity. The arachnoid mater will contain two primary structures here, the arachnoid trabeculae, which is composed of collagen fiber bundles and fibroblasts. These will anchor the arachnoid to the deep pia mater. And then the arachnoid villi, these are small bundles of arachnoid that will project superficially through the meningeal dura into the dural sinuses, and this allows for the return of CSF back into the blood. The The subarachnoid space, this is found deep in the arachnoid mater and is superficial to the pia mater, which contains major blood vessels of the brain. This area, the subarachnoid space, is filled with CSF fluid. So if you have an area where they're putting a shunt into the brain, either to drain CSF, or they might be putting a shunt in the brain to deliver antibiotics to the central nervous system, it would be through this particular space, the subarachnoid space. The pia mater, this is the deepest meningeal layer. It is the only meninx with physical contact within the brain tissue. The pia mater will follow the contour of the brain, covering delicate tissue of every sulcus and fissure. It is permeable to substances in the brain, extracellular fluid, and CSF, which is essentially the extracellular fluid of the brain. This allows for substances to move between these two fluid compartments, and it helps to balance the concentration of different solids that are found in each compartment. So here we can see the brain protections. You have the skull here, then you have the periosteal layer here, okay, of the brain. I'm sorry, the skull. Then we have our three protective layers. This is the dura mater here, and you can see the arachnoid. I'm sorry, the dura mater here, and here you can see the this layer. Then you have the arachnoid mater. This is that web-like structure right here, and then the pia mater is directly attached to the brain itself. Ventricles of the brain and cerebrospinal fluid. There are 4 ventricles within the brain, and they are linked to cavities that are continuous with the central canal of the spinal cord. The ventricles of the brain are lined with epidermal cells. They are filled with cerebrospinal fluid. The right and left lateral ventricles, these are the first and second ventricles within their respective cerebral hemispheres. They resemble a ram's horns when observed from the anterior view, and horseshoe-shaped appearance in a lateral view. The third ventricle is a narrow cavity found between the 2 lobes of the diencephalon. It's connected to the lateral ventricles by the intraventricular foramen. The fourth ventricle is found between the pons and the cerebellum, connected to the third ventricle by the cerebral aqueduct. This is continuous with the central canal of the spinal cord. Contains several posterior openings that allow CSF and the ventricles to flow into the subarachnoid space. So here we can see a lateral view of the lateral ventricles, third and then fourth. This is looking at an anterior view. You can see the ram's horn shape of the lateral ventricles. There's your third right here, and the fourth is here. So you have this fluid called cerebrospinal fluid. This is a clear, colorless liquid similar to the composition of blood plasma. It protects the brain in the following ways. It cushions the brain and maintains a constant temperature within the cranial cavity. It also creates buoyancy to allow the brain to be floating within the cranial cavity. It also removes waste and increases that buoyancy. It keeps the brain from collapsing under its own weight. Brain tissue is extremely soft. If it was laying within the cranial cavity, the actual weight of the brain would actually cause it to collapse. So one way to prevent that is you get this buoyancy where the brain is actually floating within the cranial cavity. The choroid plexuses, this is where a majority of the CSF is manufactured. Now, one thing I want you guys to keep in mind, and I know we haven't got to this, we'll get to this sometime next week with the cardiovascular system, but when you have a capillary, which are the smallest of blood vessels, what happens is some fluid actually leaves the blood and goes and mixes with the interstitial fluid. As that fluid leaves and goes into the interstitial fluid, you're essentially bringing along oxygen and nutrients. Now, what happens in the brain, because you have those epidermal cells, which in essence is creating a blood-brain barrier, they have to pass through those cells first. Now, everything that's in the blood will not have access to forming CSF. So essentially, it restricts what can and cannot pass through. What does pass through will form cerebrospinal fluid, which is essentially the fluid that will deliver the oxygen and the nutrients to the central nervous system. The choroid plexuses, these are the structures that are primarily where CSF will be manufactured. They are found in each of the 4 ventricles where blood vessels come into direct contact with the epidermal cells. You have fenestrated capillaries here, which have gaps between the endothelial cells. This allows fluids and electrolytes to exit from the blood plasma into the extracellular fluid. The choroid plexus will produce about 150 mils, or 2/3 of a cup of CSF circulating through the brain and spinal cord at any given time. 750 to 800 mils of CSF is produced daily, so old CSF must be removed from the choroid plexus to make for new CSF. And sometimes when you have people that have, say, hydrocephalus or something like that, and that puts up some pressure on this choroid plexus where it can't drain properly, they will start to build up CSF, and a lot of times they have to go in and put a shunt in to remove some of that fluid. The process of CSF production and removal occurs constantly. CSF is completely replaced about every 5 to 6 hours. Pathway for formation, circulation, and reabsorption of CSF. Fluid electrolytes leak out of the capillaries of the choroid plexus into the extracellular fluid of the ventricles. This is taken up into the epidermal cells and then secreted into the ventricles as CSF. Remember, the epidermal cells are what are going to essentially act as your blood-brain barrier here. They are circulated through and around the brain and spinal cord in the subarachnoid space. This is assisted by movement of the epidermal cell cilia. Some CSF is reabsorbed into the venous blood in the dural sinuses via the arachnoid granulations. And here you can see you're producing it here in the choroid plexuses, and then it starts to circulate around, and it gets reabsorbed back in through these arachnoid granulations here. But it is continuous all around the brain through all these ventricles, the subarachnoid space, and even down the central canal and the subarachnoid space of the vertebral column. Finally, the blood-brain barrier. This is a protective safeguard that separates CSF and brain extracellular fluid from chemicals and disease-causing organisms. It is sometimes found in blood plasma. It consists mainly of simple squamous epithelial cells or endothelial cells of the blood capillaries. They're basal lamina and astrocytes. Unique features of the endothelial cells found in the barrier. Neighboring endothelial cells are bound together by many tight junctions. This prevents fluids and molecules from passing between them, influenced by the activity of the astrocytes in the developing brain. The limited capacity for the movement of molecules and substances into and out of the cell by endocytosis and exocytosis. Substances that easily pass through the plasma membrane are able to pass through the blood-brain barrier, include water, oxygen, carbon dioxide, and nonpolar lipid-based molecules. Protein channels or carriers allow for the passage of other essential molecules across the blood-brain barrier, include glucose, amino acids, and ions. And most large polar molecules are effectively prevented from crossing the blood-brain barrier in any significant amount. While the barrier is protective, it can hinder access of medications into the brain. And because it can hinder access of medications to the brain, sometimes if you need to deliver a particular medication to the central nervous system, it will have to be administered via a shunt directly into the subarachnoid space. So here you can see a typical capillary where you get that exchange. Here you have the blood-brain barrier where you have that capillary, but now it's got to go through these additional cells. And that is the end of part 3 of the central nervous system and the end of the brain.